talk about one of several topics I've been working on recently, and this is work kind of actually dates back about 10 years, and I think I feel like we've learned a little bit every year and got to a good state where I can have got some worthwhile thing to say. Although having said that, there's not a heck of a lot that's completely new, but I think we got some pretty good ways of thinking about the problem and emphasizing some quite important things that have been kind of appreciated by some but not others. In any case, let me just tell you the general context of we want to have genome-wide models for phenotypes of interest on just based on genome-wide SNPs. And initially, when GWAS came along, we were thinking about one SNP at a time kind of testing and estimates, but in terms of estimating effect sizes, that's kind of unsatisfactory because of all the complications of multiple tagging of the same effect. And so when we're trying to have a single genome-wide model, then what we're actually trying to estimate is something a little bit different, and I'll come to that on the next slide. I say since 2010 in the notes here because that was when what became the paper was published, Yang et al. From the Vischer group in Brisbane, what became the GCTA software that was widely used. And there have been various developments since then that I'm going to be talking about. But from some point of view, it's statistically a very simple setup. It's just multiple linear regression. It's common to, just to keep things pretty simple here, I'm leaving out covariates, assume standardized phenotypes and standardized genotypes, which in classical regression, the standardizing is just convenient. It doesn't matter, but it is going to matter from what I'm talking about. But with that standardizing, the square of the effect size is the heritability of the SNP. And so heritability just provides a kind of convenient language for effect sizes. By squaring, it says we don't care about the sign. And in general, we don't. Signs are arbitrary. Those are all points I'll come back to in a moment. But because of the success of the GCTA software, and it's kind of, many of the problems with these genome-wide models are overlooked. But there are some fundamental problems that are essentially insoluble, and the best you can do is just some kind of ad hoc workarounds, and that's been the basis of a lot of kind of confusion and controversy, I would say. But the problem one is very familiar one to statisticians. In a typical GWAS setup, you've got more parameters to estimate. If you think about individual effect sizes, you've got more SNPs than study participants. So you've got P bigger than N, more parameters to estimate than the sample size. And so statisticians will know that in general is not feasible. You can't estimate more things than you've got observations. But more than that, linkage disequilibrium means that when you've got multiple SNPs tagging the same causal effect, it's impossible to deconvolve and allocate effects to individual SNPs. So that simple genome-wide model here, that is equation one here, just the simple multiple regression model, in one sense, it's a simple statistical setup, but it's complicated by these issues that make it impossible. Even just defining what the beta are in the multiple regression setup is quite difficult, because when you've got one SNP at a time, it's pretty clear what's going on. You're talking about the marginal effect of that SNP, which is all the causal effects that it tags. But when you've got a multiple SNP regression model, the underlying effects can be allocated to different SNPs in a whole lot of potentially equivalent ways. There's no unique solution, and basically we have to come up with sort of workarounds that seem to work in practice. And they have generally been of two kinds. Early on, people were generally focused on subset selection, where you'd say, okay, we know most SNPs are not going to be relevant for any particular phenotype of interest. So let's look for ways of picking out the useful SNPs, the good ones that might be tagging some causal effect, and get rid of the others. So that seemed a sensible approach, but I think broadly speaking, it doesn't work because complex traits have turned out to be complex. There's many weak causal effects genome-wide. You can't really distinguish which are the zero effect SNPs well enough, because you can have all these weak effects which collectively turn out to be important. So since that kind of landmark 2010 paper from the Vischer group introduced, what essentially was the BLUP model from being used by animal and plant breeding people prior to that, the focus has turned to, rather than selecting the relevant SNPs, shrinkage or penalized regression methods or Bayesian methods, where basically you try and regularize the problem by putting a penalty which can be a probability distribution on all the SNPs. And it's useful, the Bayesian language is kind of useful here to sort of think about what we're trying to do, because I think if you think in terms of a prior that's trying to summarize the background knowledge, that's the kind of best way of thinking about what we're trying to do, although you can do penalized regression in a non-Bayesian way. And the models I'm going to be talking about are essentially not Bayesian models. I'll get to in a moment. So there's a whole lot of complications that I have to gloss over. One of the many standard assumptions here is that because the sign of the beta is arbitrary, and for a diallelic SNP, you can swap the sign by just swapping which one is the reference allele. So because of that, most people have assumed that effect size, any distribution, any penalty function is symmetric with a mean on 0. So I guess, so let me just say, I'm going to stick to the penalty functions always being probability distribution, so we can use the Bayesian language of prior and so on. So the prior distribution is symmetric about 0 and therefore has a mean of 0. It's not obviously true, because that would be true if the reference allele was assigned randomly, but it isn't in practice. And this is one of the many things we haven't quite looked at of what's the effect of that assumption, but it's a pretty standard assumption in the field. And I mentioned in the acknowledgements on the first slide, my PhD student who'd worked on some of the stuff I'm going to be talking about today. I was trying to get him to look into the sensitivity to this assumption, which I don't think he ever got around to doing, and we should do one day. But if you're in the game of this kind of shrinkage regression with prior probability distributions, I mentioned the assumptions of symmetry of the prior, then therefore mean 0, and independence is another assumption which is not obviously true. We're not talking about lack of independence, of dependence due to LD. It's kind of assumed by definition that these betas, the tagging of the multiple tagging effect of LD has been extracted. The underlying causal variance can still be correlated. You can think about that an effect at one SNP might have implications for the effect at a neighboring SNP. But again, that's another assumption. It's almost impossible to measure that, and we think it's likely to be. I mean, suppose one thought experiment, by the way, is this idea that if you have a positive effect at one SNP and a negative effect at a SNP in high LD with it, they'll cancel each other out to the extent that you would never be able to measure them anyway. So I'm also going to assume independence on top of these other things. So if you're in this game of prior distributions to solve the regularization problem, then it's natural to start thinking about what kind of prior. And there's been a lot of effort into, and this is a busy slide, I do tend to, I'm sorry, put too much information on my slide, but this one in particular, I'm going to say none of this matters, because my kind of bottom conclusion is going to be that this is all sort of barking up the wrong tree. So there have been a lot of proposals for different prior distributions for effect sizes, and I just listed a couple of them there and plotted some of them on the right-hand side. But our view is that that's kind of the wrong thing. the shape of the prior on these effects is unimportant because basically we haven't got enough information to say anything useful about it anyway because of the too many predictors problem. And the one thing that does matter, we say, is what most other people working in this setup have ignored, and that is that they apply the same prior to every SNP. So even if you think about a prior that's a mixture distribution with several different components, you don't assign SNPs or a priori to any of those components. You try and infer that from the data. So in effect, you're assigning the same prior to every SNP. And we would say that that's really missing something really important. All the shape of the prior doesn't matter at all. But what does matter is the variance of the effect size. That's basically the only thing you can hope to say anything useful about. And the variance is going to depend on things like MAF and LD and functional annotations that you can get from those genome databases. So again, there's nothing kind of completely new in this concept. It's just that people, we think, haven't appreciated how important this is and have basically ignored this effect. So our basic position is forget about the shape of the prior, just care about the variance. So if we've got a symmetric distribution with mean 0, the variance is the next most important property that you might think about. So I introduced this notation sigma squared here for the variance of the effect size. So that's going to be important for the rest of the talk. And our claim is that implicitly in a whole lot of genome analysis, people have assumed this variance to be constant over SNPs, whereas in fact it should vary according to MAF, LD and various annotations. And that's basically the sort of gist of my talk that I'll spell out in a little bit more detail and some its implications. So it's actually quite late in the day that we finally kind of wrote down this graph here to sort of represent the sort of model structure that we're looking at. And I did find it to be very important. So we're gonna have what we call a heritability model. So we've sort of coined this term. We haven't seen other people use it, but it would seem to be an important thing to focus on. And the heritability model is a deterministic model for these variances. So although I've talked about priors and the Bayesian language, it's fundamentally not a Bayesian setup. So the effect sizes are random variables, but we basically don't care about them. They're sort of nuisance parameters, if you like. We only care about this variance parameter. And then we're going to model this variance in terms of a heritability model which is a function of MAF, LD, all those kind of things that you might know about a SNP a priori before you do your GWAS, or just from the genotype data of your GWAS. Now, that doesn't solve all the problems. But it does help with a lot of things. So I've already mentioned that the squared effect size in our setup with standardized phenotypes and genotypes, the squared of the variance is the heritability. If those effects were correlated, the heritability wouldn't be just a sum of the squared effect sizes. So but under our assumption of at least uncorrelated effects, you can just add up the squared effect sizes to get the heritability of a set of SNPs. So our kind of basic framework is to estimate the heritability. So I just use this H squared symbol as shorthand for the word heritability. Estimate the heritability model parameters. Then we get the fitted values for the variance term, which is, and that is the expectation of the beta squared term. So in other words, it's just a mean model if you think you're modeling beta squared rather than betas. And so then you can add up over a set of SNPs. And here's another really important thing that we haven't explored in great detail yet. The estimate of the heritability for a single SNP is still going to be pretty useless for all those same reasons that I said at the start. But your estimate over sets of SNPs can be pretty good, provided that the LD effects don't mess things up too much. And so that basically means we need the SNPs to be in contiguous blocks without too many boundary effects. And we haven't really looked into in great detail when, but we'll give some examples, and we give estimates of standard errors on our estimates that show that you can estimate heritability reasonably well for sets of SNPs down to, you know, something like 0.5% of the genome or something like that. So I mentioned the sort of seminal paper from the Vischer group in 2010. Another of the big advances in the field was in 2015 with LD score regression, the Boston group, Bullock, Sullivan et al. So this is a picture just to sort of illustrate the general idea. On the left, every dot is a test statistic, and its x coordinate is a measure of LD for that SNP. And so the basic idea is that the more LD there is, the more causal effect that's being tagged, and therefore the greater the mean value of the test statistic over the genome. And so that's why this line here has a positive slope, and the slope is related to heritability. The effect is much easier seen if you bin the SNPs, and so that's the picture on the right, and I think that's corresponds roughly. These are our own plots. This one on the right is similar to the plot in the 2015 publication. I don't think they showed the one on the left. It's a bit more a kind of honest description, so you know there's relatively little variance explained in the test statistics, but nevertheless you can estimate the slope reasonably accurately, and from that you can get heritability. And this turned out, you know, for all the advantages of using summary statistics rather than individual level data, this turned out to be a big step forward and has been widely used. Here's just a brief summary of the LD score regression model. So I'm assuming many people are familiar with this, that it's a model for the test statistics. So that's that equation 2 at the top here. It's just a simple linear regression where the slope depends on this LD score. And if you write down the main, so this A is an intercept term, which I can discuss a little bit later, but the main main interest is in the slope, which is spelled out more fully in this next displayed equation. And you can see that the slope is assumed to be a heritability term times the LD score, which is some measure of LD, just adding up the R-squareds in some region. And there's a few more details there that I will skip over. But what I want to focus on is that this heritability term is the same for every SNP. And so we refer to that as the uniform heritability model. And you know, I've already made this point that the main gist of our talk is to say that this uniform heritability model crops up as an implicit assumption in all kinds of genome analyses, and makes them all suboptimal because it's a terrible model in terms of what we really know. And so specifically for LD score regression, so here I mentioned, in addition to LD score, this uniform model is implicit in so many other software. We just listed some of them here, but the actual software that we found that implicitly make this uniform heritability over SNPs assumption, the list just goes on and on and on. There's a huge amount of them, and we can do better. And again, our core idea is we can do much better in the models underlying all of these software by improving the heritability model. And so now you have to compare this term here with the original LD score, and the difference is that we now have an explicit variance term for every SNP. The sigma squared J was a constant in the original LD score model, and we now allow that to be SNP specific. And then and again, you can't estimate a degree of freedom for every SNP, but if you express it as a function of MAF LD parameters, genome function parameters, then you've got an estimable model, and you can get estimates of this sigma squared J and use it to improve LD score regression. And I just note again this point about focusing on the distribution of the betas is a bit of a waste of time, because in this LD score regression, the betas don't even appear. They don't even exist in the model. It's only this variance term. So it's just the variance of the effect sizes that we model in terms of MAF and LD and all these other things. All right, so that's the introduction to the main ideas. Now we just move on to some details about specific models. And when we came at this starting in a 2012 paper, we focused on the effect of LD. And if we were now doing it again, we would start with MAF, because MAF and LD are quite closely connected. Of course, they're and MAF is a simple one parameter, one number parameter, which is kind of, relatively easy to model, whereas LD is a kind of more, is a more complex phenomenon. But also we got into a bit of an argument with the Boston group, when in fact we got into arguments with everyone, the Brisbane group and the Boston group, but specifically with the Boston group behind LD score. There was this sort of interesting idea that their intuition based, the intuition underlying LD score was that the more LD there is, the more heritability, the tag. And that's what LD score is kind of based on. When we looked at this, we saw that the more LD there is, the less heritability. And so there's a couple of ways of thinking about that. One is that heritability gets overestimated because of multiple tagging in certain frameworks. But another idea is that if you think about a set of SNPs in perfect LD, they only have the same variance as a single SNP. And so they only have, they have reduced possibility for function compared to less correlated SNPs. So we seem to be going in completely different directions from the Boston group. They were saying more LD means more heritability. We were saying more LD means less heritability. We can't both be right, surely. But in fact, we would now say that we are both right. And it's got to do with whether you do or don't extract out the effects of multiple tagging, because what the Boston group were talking about is that because of multiple tagging, the apparent heritability, the marginal effect of individual SNPs goes up with LD. But the underlying contribution, once you extract out the effects of multiple tagging, the underlying heritability per SNP is reduced with more LD. Both of these things are true. In the simplest setups, they cancel each other out exactly, but in reality, they don't cancel each other out exactly. They're different things. They're both correct. They both have to be modeled. But anyway, let me come back to, I'm going to present this now focusing on MAF rather than LD, because the LD, you get into these complicated arguments. But if you're going to say, what's the relationship between MAF and heritability, then this model 4 is a pretty natural, simple one. You've just got a power term here based on the MAF, and then the P1 minus P form is convenient for various reasons. The 1 plus alpha here is because the 1 kind of cancels out the standard scaling when the genotypes are standardized. And I've got a little bit of detail here. As I said before, I do tend to put too much on my slides, but I just, in order to discuss the relationship between MAF and effect size, it's useful to think about unscaled genotypes, whereas in fact, most people do standardize and therefore introduce some kind of scaling. Maybe I'll just move on to the next. So if we're going to adopt this model, and we think it performs pretty well, but of course it's not obvious that this is the right model, there may be better ones, but it's a simple 1 parameter but nonlinear model relating MAF to heritability. And this uniform heritability model that I've been describing and criticizing corresponds to alpha equals -1, which basically just says if you put alpha equals -1 here, then the MAF term just disappears. And that's the uniform model that says the variance is the same for every SNP. This is very, very widely assumed in human genetics. We mentioned it's implicit in GCTA, it's implicit in LD score regression, it's implicit in all these other prediction software. But it's a terrible model in terms of looking at data. It's hopeless. It's way, way off the truth. I'll show you a bit more. But while the human geneticists were typically assuming alpha equals -1 in this model, the animal and plant breeders were often assuming alpha equals 0. So in other words, they were just assuming that effect size distribution was uniform over SNPs rather than heritability being uniform over SNPs. So it's then an interesting empirical questions of which of these is right, or is it something else? And then to cut a long story short, we found that -0.25 fits pretty well So somewhere between these two standard assumptions. And the diagram here on the right is from our 2017 paper, where with the blue line showing so the plot is showing the relationship between expected heritability or average heritability and MAF. The blue line is the uniform heritability model. The gray bars represent empirical data over many GWAS, and we just partitioned into two parts here, less than 10% and more than 10%. So you can see that the alpha equals 0 assumption of the animal and plant breeders is not too bad, but minus 0.25 is better, and the uniform model is hopeless. Again, there's sort of nothing new here. Many people would appreciate that heritability does vary with MAF. It's when you state it, it just seems obvious, and yet that assumption is implicit. And there's a sort of long story of why it's implicit in so many analyses, and it's because standardizing genotypes is a kind of natural thing to do, and many people did it, and then it became a kind of hidden implicit assumption that the heritability was the same, but in other words, that the standardized effect size was the same over SNPs. So here is some empirical data from the UK Biobank, 14 traits picked out, and for this data set, the average alpha turns out to be around about minus 0.18. Minus 1, which is the value widely assumed in GCTA and everything else, is way off the scale. It's way down here on this plot. We've never seen any phenotype for which the estimate, an empirical estimate of alpha, is anywhere near this minus 1 value that's pretty widely assumed in many human genetics analyses. So this alpha parameter, this relationship between MAF and heritability, is often interpreted as a measure of negative or purifying selection. Alpha equals 0 says that the unscaled effect size variance is constant. Alpha equals minus 1 says that the scaled effect size, so in other words, the heritability, is constant over SNPs, and our recommended average value of minus 0.25, as I said, that our estimate was minus 0.18 for those particular phenotypes. So it's somewhere around about that. Because it's a nonlinear model, it's actually hard to estimate alpha very accurately for big data sets. But that corresponds to some weak negative selection. And if you look at those 14 traits, they're all consistent with weak negative selection. For some of them, neutrality is not rejected. And then there's that sort of disappointing fact for people in this room that of the traits that we looked at, the one that nature seems to hate the most is getting a college education, that it sort of shows up as being the strongest negative selection against that trait. And of course, you can kind of speculate as to why that might be, but I'm not going to take any of that too seriously. There's many other selection models that can affect this alpha parameter. So it's only it's only useful as a sort of loose interpretation. But yes, going back here, you can see the college education term here is a little bit more negative than the others, but they're all pretty broadly consistent with the sort of weak negative selection model. So my argument has been that there's this uniform heritability assumption that's implicit in many analyses. You can do much better by replacing that with a better model. So far, I've just showed you one model with just one parameter, which is the MAF term. And if you adopt our default value of minus 0.25, which we say works pretty well over many phenotypes, then it has no extra parameters at all. It's still just the same number of parameters as the uniform model. But so now I'm going to show you that can, even just that simple model of relating the MAF to the heritability, can make big gains. And this is a paper which is on medRxiv at the moment, but under review, and we hope will be out in a good journal soon. And this is doing gene-based association. So you define genes however you like, and then we want to do an association test based on basically the estimated heritability of the gene. And there was some software from Listgarten et al. Doing that in the software called FastLMMset. And what we've done is pretty similar, but again, they implicitly assume the uniform heritability model. So obviously our generalization is to put in, because of the computational demands for this particular analysis, just this extra term of having the relationship between MAF. So having heritability depend on MAF, expected heritability depend on MAF was our main step forward. But we could also do this. The original paper was only for individual level data, whereas we now do it for summary statistic data. So we used that MAF scaled model that I just introduced with the default value alpha equals -0.25. And that's a simple enough model that you could actually derive an approximation that works for summary statistics. And so we could potentially do better with better heritability models. But this slide is, and you know, see our paper on medRxiv to get more details. But basically, we do better than every other gene-based association test that we could find to compare with. And just from this one insight of modeling the relationship between MAF and heritability. So that's us here, and that's all the other models. Obviously I can't go into details here of exactly what we did. This is for empirical data, and we've done some work to show that our model has correct type 1 error. And so therefore empirical discoveries, we hope, should correspond to power. This plot in the top right is comparing our gene-based association test with the first light blue bar is the min p, just doing your gene-based association based on the best p-value. And so you lose a lot with the same sample size. If you increase the sample size by 20%, you still don't quite catch up. If you increase the sample size by 40%, you do better. So roughly speaking, modeling the MAF relationship corresponds to about a 30% increase in sample size. And we'll find that is roughly true for some of the other analyses I'm going to show you. And then finally, on the bottom right, it's just showing the number of discoveries for different alpha values. And again, we found this -0.25 being the best of the ones we considered. But again, we can't estimate that accurately because it's a non-linear relationship. We just have to basically turn the handle on the computation for specific alpha values. And so we tried these alpha values, and alpha equals -0.25 turned out to be the best again. Oh, sorry, I see I've got the low battery warning, and I haven't got my laptop plugged in. It's in my back. I'll just run and get it. Sorry, 2-minute break while I plug in the battery. Right. Oh, I just got the, so I just need to put that into a wall socket. Where's that? Yeah, there it is, US one. Complicated adapter. Where is the power source? Oh, here it is. Yeah. Right, sorry about that, I'm back. So one of the big advantages of LD score regression was the model is so simple that you can put lots of predictors into the model. And so when the Boston group developed LD score, they saw straight away that they could put a lot of predictors into the model, and we were sort of in some sense in competition with them, and they got a big head start at that time by putting in lots of genome annotations into their model. So that was what they originally called LDSC or stratified LDSC, which basically just meant putting in a lot of binary predictors into the model, where the binary terms were whether or not a SNP was in a certain genome class, like coding SNPs, intronic SNPs, all kinds of other things. I'll show you the functional categories they used in a subsequent slide. So they developed this baseline model with 53 parameters. Now, for statisticians, generally 53 parameters is a lot, but because you've got of the order of millions of, or hundreds of thousands of sample size, it's actually not a large number of parameters. And then in a later paper, they, instead of just having binary predictors, they extended it to continuous predictors, which could be various continuous measures of LD. And then they came up with this baseline LD model. And meanwhile, we were proposing certain other models. And so, this question of how you do model comparison for these summary statistic models comes up, and we had various ways of trying to do that. But eventually, of course, for statisticians will want to have a sort of likelihood-based comparison. So Matthew Stevens and colleagues have published a likelihood for summary statistics, but they're focused on the likelihood, the parameters being the effect sizes, and it was only for individual level. Sorry, it is for summary statistics, but it was very difficult to use, and we couldn't use it easily for model comparison. We came up with an improved likelihood for summary statistics, not for the original GWAS data. And I just want to advertise that we did this, but I won't go into any more details here. And we did some work to show that it worked pretty well, but I'm just going to skip over that in the interest of time. And so we've got all these different models. I've summarized some of them. I've mentioned some of these models along the way. The uniform model, our LD-based model in our 2012 paper, we've moved on from that, but it's just there for comparison. The GCTA people sort of took on board that LD and MAF matter, and they came up with a stratified model. And then there was this baseline and baseline LD from the Boston group that had all these genome annotation predictors in. And when we could do good model comparison, not surprisingly, we found that this baseline LD model from the Boston group with 75 parameters was the best and better than anything we had at the time, but we kind of turned defeat into victory by basically stealing their model and improving it, because they had a sort of clumsy way of dealing with MAF of just 10 MAF strata, each with its own parameter. And instead, we put in our one parameter power model that I showed you earlier. And we did also put in our LDAC weights, but basically, once you've dealt with MAF correctly, the LD effect can be dealt with just by thinning out the highly correlated SNPs we've subsequently found. So we actually moved away from the LD weights that we were promoting a few years ago. So the baseline LD model was the best in about 2019 or 2020, and then we improved a little bit, reduced the number of degrees of freedom, but with a better fit. And so we come up with this BLD LDAC model. We call it, a bit clumsy, and we've actually moved on from that since, so I don't want to spend too much time emphasizing this particular model. But this is now an enhanced version of the graphical structure that I showed you earlier, where all of this stuff at the bottom was in a single term that I called heritability model, and here it is now expanded a bit more. But so these are the genomic annotation predictors. These are our LD weights. These are the MAF terms. They all go into this variance of the effect size, which is the expected heritability, and then that can go into subsequent models. And one of the things you might want to do is prediction. So I mentioned the gene-based association, and we got big gains by just a very simple heritability model. That BLD LDAC model that I just proposed was used to improve prediction in a paper from my colleague Doug Speed. So this is a paper I'm not an author on. I was just loosely, distantly involved in this, but they did something pretty clever of took all the available prediction algorithms they could find, both for individual level data and for summary statistic data, and pretty much all of them could be improved just because they all relied on the uniform heritability model, and by replacing that with a better heritability model, you could improve them. So that's the bottom line. I'll give you a few details. This slide is for methods based on individual data. And what Doug first did was re-implement all these methods within his LDAC software. So I've mentioned the LDAC model. There's also the LDAC software, which Doug Speed maintains. So this LDAC Bezier Predict is the Bezier prediction software in the LDAC implementation of it. And one reason for doing that was Doug was able to speed it up. For these analysis, his version was 50 times faster than the original version. And his version for several of these methods is faster and more accurate than the Bolt Predict is from another Boston group. And his version of it was better. This is the LDAC implementation of ridge regression. This is the LDAC implementation of the lasso. And what's going on here is the horizontal black line is a kind of classical PRS just a standard kind of one. The red bars are the original published versions of each of these methods. Well, for the lasso and for the ridge regression, they're just sort of standard methods, but for Bolt and Bézard, they're published versions. So the red is what the original versions could achieve, and then the light blue is the improvement you get with a simple heritability, one parameter heritability model, and then the dark blue is you do a bit better again by having this 65 parameter heritability model. And again, 65 is a lot of parameters. In some sense, it doesn't matter because we've got huge numbers of observations. And again, roughly speaking, the improvement in prediction corresponds roughly to about a 25% increase in sample size. For the individual level data, we found that Bolt LMM was the best method. And so you can see this red bar is the highest of the red bars, but we could improve it again by improving the heritability model. I keep saying we, as I said, this is not my work. And this is similar kind of conclusions, but now for summary statistic data. And so we could look at many more phenotypes here. The methods we show here, I think we looked at more, but there's lasso sum, SBLUP, LD-PRED, and S-Bézard. So all of them assume the uniform heritability model, and the light blue is showing the improvement you get from a simple one parameter heritability model, which incorporates the thing and the MAF term, and then the dark blue is the further improvement. So on the right is just binning all the dots on the left to see them more clearly. So anything above 0 means we got an improvement. So it's actually not strictly true that we improved in every case, but we improved in 223 out of 225 traits. I mentioned earlier that these functional annotations, and so I'm going to look at them a little bit more closely now. These were the functional annotations proposed by the Boston group. I think it dates back to their 2015 paper. And this is the percent of SNPs that fall into that functional class. So 1.2% of SNPs from the UK Biobank. All our data analyses pretty much are UK Biobank data. 1.2% of SNPs are coding. This conserved category is interesting. This was conservation amongst, I think, 29 placental mammals, and I can't remember the exact definition of conservation, but to give you an idea, about 2.4% of SNPs were classified as being conserved. So an interesting question is, which of all these annotations, you can see all the other annotations here, transcription start sites and 3' UTR and all that sort of thing, which of these is going to be most important in terms of predictors of heritability? Any guesses?
Super-enhancer.
Yeah, well, that's a good guess, but no, it's not going to be the one. And another guess I got when I presented this previously was coding. That's an obvious guess, but that's not going to be the important, and it just comes up on the next slide. So far and away, that conserved category turned out to be the best predictor of heritability. And so I'm showing two things here. The top plot shows the heritability within the class relative to a kind of baseline genome-wide average heritability, and how you define that is a whole argument as well that I won't go to in any detail here. And conserved over on the left is up over 7, so that says there's 7 times more heritability in that 2.4% of SNPs that are conserved. There's 7 times more heritability than you would expect under a sort of genome-wide null model. And so coding, by that definition, coding is the next most important, then 5' UTR, then transcription start sites. But basically, we've got a multiple regression model. And so this is the sort of marginal importance in the top plot, marginal importance of each of these categories. This is the joint effect. So these are basically the estimates of the parameters from the multiple regression model with our 65 parameters. These are the 24 corresponding to 24 genome annotations. And again, conserved comes up as way the most important. And because this is jointly fitting these terms, that means conserved is important after adjusting for coding, 5' UTR, transcription start sites, and everything else. And so I don't really have a good explanation of why conserved is so important. but it's not simply because the conserved regions are coding, because we've already got coding in the model, and conserved still comes out as super important. And in fact, you can see with our 65 parameter model, although you can estimate 65 parameters easily enough, because of complicated correlations amongst the parameters, your marginal precision of the estimates is poor, you can see for many of these. And so that's the last bit of the talk that I probably won't have time for, because I'm a bit running out of time, is that we want to move away from that 65 parameter model and reduce the number of parameters quite a lot. But the other interesting things, there's a whole story around DNA's hypersensitivity sites. In, I think, the 2014 American Society of Human Genetics, the Boston group announced an estimate of very, very high enrichment of heritability in DNA's hypersensitivity sites. You know, exactly how that's defined, I need to look up, but it's about 16% of the genome are classified as DHS. And we reanalyzed and found a very different result from them, and they never really explained their original result. They just stopped talking about it. So I think that means they agree with us. But interestingly, the regression parameter is negative. So after adjusting for everything else, DHS sites have less heritability than you would expect, which is a bit of a puzzling observation. Now, they do marginally have more heritability than average. That's because there's a lot of other interesting stuff in DHS regions. But once you've adjusted for all those other things, the fact that it's a DHS region reduces the amount of heritability, which is a bit bizarre, and I don't have a good explanation for that. So I just got a page of conclusions here. I've mentioned, already talked about some of these, and I'm short of time, so I'll skip over this. The enrichment of heritability is relative to a baseline, and how you define the baseline is, of course, a bit open to discussion, but it might in particular have something to do with why conserved comes out as important. The importance might be exaggerated because there's low variation in these conserved regions, sort of by and that may affect the baseline, which kind of inflates the importance of the conserved region. But even after allowing for that, that would only be a sort of factor of 2 effect at most. So conserved is still very important. And this is looking at the MAF parameter alpha that I talked about before, but now specifically for each of these regions. And it turns out, consistent with the idea that most of these functional regions are important, if you think of this as a selection parameter, so the selection effect is stronger inside these regions than outside for every one of these except repressed, where it's the other way around. But I've also left out here the non-significant regions, and conserved is one of them. So in other words, this MAF parameter, interpreted as a measure of negative selection, isn't significantly different within inside and outside conserved regions, which seems a little bit puzzling, and we don't have a good explanation for that. So I'm now near the end of my time, so I should try and finish up, but this is some work that my recent PhD student has been doing in developing a better 10 degree of freedom heritability model, and just to show that it works about as well as the 66 degree of freedom model, and gives you, because it's easy to handle, there's a lot more analysis you can do, like the enrichment of heritability in EQTL sites for different phenotypes, looking at male versus female. All of these analyses have been done before by others, but we think our analyses are better. Maybe not always significantly better, but often substantially better because of the better heritability models. So that yes, there's a lot more work to come out of that. And in fact, even just a 3-parameter heritability model works almost as well as the 10-parameter one, but I don't have time to give you any details. So the conclusion is that a lot of existing genome-wide analyses that will within this genome-wide regression framework, including GCTA and LD score and many others, you know, LD bread and all these other Bayes-R and all these other prediction software, they can all be improved by focusing on this heritability model and putting in better models for the expected heritability. We got some interesting results from our new analyses. I don't want to promote too much any of the specific models because they're probably better models. They're all these predictors of function that have been, there's been a lot of them coming out. Recently, I mentioned this one, Macy from Lee et al. Just come out in the last few months, which looks pretty good. So there are all these new predictors, composite predictors of heritability, each of them, based on multiple inputs. And probably with those, we can come up with maybe just a 3 degree of freedom heritability model, which will give all these gains in inference that I've been talking about. So a lot of this discussion is summarized in a recent bioessays paper, and that turned out to be, it's a sort of review essay type of paper. That's my student's PhD thesis I referred to. This is the gene-based association one, which is on medRxiv, and that's the prediction one from Doug Speed and his colleagues in Denmark. So I'll stop there, and thanks for your attention. Let's see if I can, if anyone's, I have to unhide something. Yeah. Now, why is that not there? No. I said unhide the video panel, but I can't see it. Any case, is that? Now there it is. Yeah, there we are. All right. So it is now, the hour is up, so some people may need to go, but I'm in not any hurry, so I'm happy to stay and answer a few questions if anyone wants to just unmute yourself.
Yeah, please do that. So what are these 10 new parameters?
I do have that somewhere else that I could just get them up, and so basically the student did a new search. So for up until recently, we've been using those same functional annotations that the Boston group introduced 6 or so years ago. So they're getting somewhat out of date now. But the student just did a new search based on trawling through genome databases and came up with 335 potential predictors of heritability, and then did because the data sets are big, we had to do a fairly limited kind of stepwise selection process to pick out the 10 best ones. And I can, and again, conservation-based ones turned out to be, so there were 2 or 3 that were related to conservation. If you just give me a minute, I might just find the rest of them. Yes, yeah, MAF is by far is very important, and in fact, all of our predictors are scaled by that MAF term now. So the effect of MAF is kind of pervasive in all of the things, because it's sort of a given that that's an important one. I had that.
You see that check button you've got?
Yes. Yeah, I'm just having trouble seeing my own screen. Yeah, that's better. Right. It's about the first time I've seen a speaker fleet. Thanks, Joe. All right. So I don't think there's any new, but I have got this. Let me just. I can. I don't think I can pick out the. Just find in a minute the predictors in the new heritability model, the top 10. So these are some of the correlations amongst the. Yeah. Sorry that this is not shared with those online, but I can just read out. So they're the 10 predictors. So there's the MAF term, and some of these are, require. So CPG content turned out to be an important predictor. The H3K9AC marker turned out to be important. So this fast cons is another conservation-based predictor. This is primate conservation-based predictor. This is an age of alleles. This ASMC score is an age of allele type of thing. So, but yeah, again, the new maybe I can, I don't know what, whether I'm succeeded in sharing that or not, sorry. But
So
yeah.
what's happened to LD? Summarize what you've done.
So basically, there is this inverse LD score. So instead of our LD weights that we were promoting some years ago, we've kind of backed away from those as being a bit complicated and just basically replaced them with an inverse which is basically the same thing as in LD score regression. The inverse of that is a predictor of heritability, this is showing the importance in terms of the standardized. This is the number of standard deviations of the regression coefficient, and so you can see it's very high for that. So LD is an important predictor. MAF looks like it's less important here, but that's a bit misleading because all of these predictors are weighted by the MAF term. So MAF is definitely the most important. LD is very important. Conservation is important. Yeah, they're all good predictors of heritability, and that can go into all of these analyses of prediction. So
how much of that is because these are probably pretty well measured, as opposed to?
Sorry, could you just speak up a little?
How much of that? How much do you think that the ability of those parameters as predictors is sort of on top of the heap because they're probably well measured, as opposed to maybe some of the other things that might be around that are kind of more squirrely?
Yeah, absolutely. So thanks, Ellen, for the question. So just I have no kind of confidence at all that any of these predictors is, any of these models is going to be the best in any kind of absolute sense. There's a huge model space out there, and there's a ton of data, and so yes, the accuracy of measurement is one thing, and also
some data is better than other data.
Yes, and new genome annotation data is coming out all the time, and again, these composite predictors of function. There's quite a lot of those coming out. We kind of feel that for our purposes, it's better going back to the raw data rather than using a composite predictor, but maybe you can come up with a good model based on some of these composite predictors that are being proposed recently. That so any of these models that exist for predictors of function could potentially be a predictor in our heritability models. Any other questions?
So thinking about SNP arrays versus whole genome sequence data, the one question is, can you just apply the stuff directly to whole genome sequence? And the other is, if you're analyzing SNP arrays, do you impute
Can you repeat the question?
Yeah, sure. So what about SNP data? So this reminds me of one of the many details that I didn't mention, was that most of our work until recently followed the convention that was in place up until fairly recently of only including SNPs with MAF above 5%. And the most recent work in the last couple of years, we lowered that threshold to 0.5%. And we have had a stab at trying to include rarer SNPs, but generally none of the results I showed you involve SNPs with frequency below 0.5%. So the rarer variants that you can pick up with sequencing data are not included in these models. And of course, they are potentially important, and that's an obvious direction to go. But it's harder to, it's the usual kind of problems about small sample size means it's hard to estimate parameters and all that kind of thing. But definitely we now got sequencing data on a scale where we could do that, and we just haven't done it yet systematically, and that would definitely be a good thing to do. And obviously, so you've got a whole industry of predicting function for the very rare variants, and then there's that category now of sort of moderately rare below 0.5% and set down to the sort of singleton variants, and all of that is not included in our models yet. So that's as I said, an obvious direction. It would be great to be able to have a grand unified model of everything from the extremely rare up to the common, and we don't have that at the moment. And so, for example, we tried to look at, does the MAF relationship continue to hold as you go to rarer and rarer variants? And it doesn't seem to. Our estimate of that alpha parameter gets shrunk towards zero as you get to rarer and rarer variants, but we don't know whether that's a sort of real effect or it's got something to do to a sort of regression to the mean effect because of the reduced information content in the data. I don't know. Any other questions?
How would a super rare variant contribute much of anything to the population level heritability? Because hardly anybody has it.
Yes.
So
well,
I think that doesn't make sense to think about doing that. To me, the super rare things may be interesting from a biological point of view because they might lead you to biology, but for prediction, they're not going to do anything.
I guess so. But if you're thinking of applying, and so the argument is that very, very rare things are kind of useless for prediction in population-based prediction. But all of us have got some very rare variants, right? And
true.
If you can kind of at least conceptualize a very, very thorough prediction model that when you put in my genome, it can get some estimate for the extremely rare variants and go into my prediction. That seems to me like a sort of impractical at the moment, but at least conceptually feasible. That in the future we could have estimates of lots and lots of extremely rare things. But yes, I mean there are things. Certainly some of these rare variants have such big effects that once you've got that effect, nothing else matters, and so that makes the rest of the prediction model kind of redundant. I agree, but we can definitely go down well below 0.5% with the sort of big genome-wide sequencing data sets that are now becoming available. We can go down to much less than 0.5%, even if not down to, even if the getting down to extremely rare would be impractical and not very beneficial, as you say.
So have you put a population structure related and all that stuff into this?
No, that's all part of the actually, and we did recently give a one-day short course, which was kind of an expanded version of this stuff where we went into more detail on all of these things, and we did so that's a little subtopic that I skipped out because, as you saw, I already had too much content anyway. But just briefly, the LD score was introduced as a way of measuring confounding, and because that A intercept term in the LD score model was interpreted as their genome-wide effects that don't depend on SNPs, and so that's due to population structure. And so they were proposing this as something better than genomic control, which was popular at the time, but now seen as too conservative. But they never really showed that it worked, and we sort of followed in their footsteps for a bit until in our 2019 paper in Genetic Epidemiology, we showed that actually that the A intercept term doesn't measure the effects of population structure very well. That although on a broad scale, on a fine scale, LD is population structure does affect LD, and so it does mess up the LD score regression. So LD score and all these other versions of LD score regression, the generalized versions I've been talking about, all rely on population structure effects having been eliminated in the primary GWAS analysis underlying the test statistics. So putting in that intercept term might help a bit, but doesn't give you any guarantee of protection from population structure. So we haven't solved the population structure problem, but we have to rely on the original GWAS authors to have dealt with population structure adequately.
Well, we should probably stop. So thank you all for coming. Thank you, David. That was comprehensive.
Thank you, Bruce.
A lot of food for thought.
Yeah. It was interesting. Thanks.
Sorry?
That was very interesting. Thank you.
Thank you, Alan.
I couldn't get down there.
Yeah, it's a shame. Yeah. All right.
Yep. Thanks, David. I couldn't get down as well, but it was very interesting stuff.
Thanks.
Look forward to reading more about that.
Thanks, Ken.
Cheers.
All right.
Thought I was recording. I don't think so. I seem to have lost.
It says recording here.
Does it?
Yep.
Can you press the stop